Welcome back to Immunopolis Central Laboratory. Today, we will explore how antigens and antibodies interact and how these interactions underpin serological tests used for diagnosis, disease monitoring, and research. Here, just as puzzle pieces, antigens and antibodies float, waiting to find their perfect match. This intricate dance of identification is the essence of serology, the science that reveals who binds to whom. Whether inside the body, in vivo, or in a test tube, in vitro, the precise interaction between an antigen and an antibody is paramount. Before delving into the diverse forms of serological reactions, we will first establish the fundamental principles governing antigen-antibody interactions, setting the foundation for everything that follows. A strong antigen-antibody reaction depends on a very close fit between the antigen's epitope and the antibody's binding site, just like a lock and its key. This complementarity explains why antibodies are highly specific for their target antigens. While the lock and key analogy suggests a simple fit, the reality is far more complex. An antibody doesn't just snap onto an antigen, it's held by a symphony of forces. Individually, these non-covalent bonds, such as hydrogen bonds, ionic forces, hydrophobic interactions, and van der Waals forces, might seem weak. But collectively, they create an incredibly strong and highly specific attachment, ensuring our immune system targets precisely what it needs to. A perfect balance, where every antigen finds its antibody, is mandatory for the visibility of antigen-antibody interactions, forming an intricate, stable structure. This is the zone of equivalence, where antigens and antibodies link together to create a visible lattice, much like a crystal bridge. This optimal ratio leads to the maximum visible reaction. If either antigen or antibody is in excess, visible reactions decrease. In the laboratory, monoclonal and polyclonal antibodies serve as powerful detection tools, each with a different purpose. Monoclonal antibodies, because they recognize a single specific epitope, are used when high specificity is required, such as in enzyme-linked immunosorbent assays and flow cytometry, where false positive reactions must be avoided, while polyclonal antibodies, which bind to multiple epitopes on the same antigen, are preferred when strong signal and high sensitivity are needed, such as in immunohistochemistry, Western blotting, and immunofluorescence, where proteins may be present in low amounts or altered in shape. Together, these two antibody strategies allow laboratories to precisely identify pathogens, measure biomarkers, visualize proteins in tissues, and even design targeted antibody-based therapies. Generally speaking, direct serologic tests use known antibodies to detect unknown antigens and are mainly used for rapid screening. Indirect tests use known antigens to detect antibodies in the patient's serum and can be either qualitative or quantitative. Quantitative antibody assays measure the titer, which reflects the amount of antibody in serum. The patient's serum is serially diluted and added to fixed amounts of the target antigen. The highest dilution that still yields a positive reaction is recorded as the antibody titer, which is useful for monitoring disease progression. After we have learned the basics, let's delve into the various modalities of antigen-antibody interactions. In this agglutination arena, antibodies act like skilled grappling hooks, locking onto particulate-insoluble antigens displayed on red blood cells, bacteria, or artificial particles and pulling them together into visible clumps, revealing the presence of the target. In direct agglutination, antibodies bind naturally to antigens already present on cell surfaces, such as red blood cell antigens, resulting in rapid, obvious clumping. In passive or indirect agglutination, soluble antigens are first coated onto carrier particles so that antibodies can cross-link them into lattices. A good example of passive agglutination is latex bead agglutination, which uses antibody-coated beads to detect microbial antigens in fluids such as the cerebrospinal fluid. Agglutination reactions are further classified into slide and tube agglutination tests. Slide agglutination is a rapid, qualitative method performed on a glass slide that enables quick antigen detection through visible clumping. It is widely used for ABO blood group typing and for the identification and typing of bacteria, making it an invaluable tool for rapid, bedside and laboratory screening. Conversely, tube agglutination is a more sensitive and quantitative method performed in test tubes. It is used to detect and measure antibody levels, or titers, by observing agglutination across serial dilutions as previously illustrated. Interestingly, during serological testing, Accurate interpretation of antigen-antibody reactions depends on maintaining an optimal balance between the two components. If either the antigen or the antibody is in excess, the reaction's visibility is compromised. The prozone phenomenon refers to a false negative or weak agglutination reaction caused by an excess of antibodies, which interferes with lattice formation. In contrast, the postzone phenomenon occurs when there is an excess of antigen, leading to insufficient antibody cross-linking and, consequently, weak or negative reactions. Both phenomena are most commonly encountered in tube agglutination and precipitation tests and can be resolved by serial dilution of serum, for prozone, or adjusting antigen concentration, for postzone, to restore the optimal antigen-antibody ratio and reveal true positivity. Hemagglutination is a type of agglutination in which antibodies bind to antigens on the surface of red blood cells, causing the RBCs to clump together into visible aggregates. It is widely used in blood group typing, antibody detection against RBC antigens, and in some viral assays in which viruses can bind and cross-link red blood cells. In blood typing, red blood cells are mixed with anti-A and anti-B sera. If agglutination occurs, the corresponding antigen is present on the RBCs. This is essential for safe blood transfusion. This illustration demonstrates a BO blood grouping by slide agglutination, in which red blood cells are mixed with specific antisera and observed for visible clumping. Type A blood shows agglutination with anti-antibodies, indicating the presence of an antigen on the red cells, while type B blood clumps with anti-B antibodies, reflecting B antigen expression. Type AB blood agglutinates with both anti-A and anti-B, as it carries both antigens, whereas type O blood shows no clumping with either antiserum, confirming the absence of A and B antigens. The visible clumping indicates a positive antigen-antibody reaction and serves as the basis for rapid ABO blood typing in clinical practice. Another application of hemagglutination is the Coombs test. The Coombs test, also known as the antiglobulin test, is used to detect immunoglobulin G antibodies or complement components that are either bound to red blood cells in vivo or present freely in serum. The Coombs test is commonly used to diagnose fetal-maternal Rh incompatibility or autoimmune hemolytic anemia. There are two types of the Coombs test, direct and indirect. The direct Coombs test detects immunoglobulin G antibodies or complement proteins already bound to a patient's red blood cells in vivo. In this test, the patient's red cells are washed to remove unbound serum antibodies and then mixed with anti-human globulin. The appearance of visible agglutination indicates a positive result, confirming that the red cells are coated with antibodies or complement. On the other hand, the indirect Coombs test detects free, circulating immunoglobulin G antibodies in the patient's serum that bind to red blood cells. In this test, the patient's serum is first incubated with test red blood cells. After washing to remove unbound antibodies, anti-human globulin is added. Agglutination indicates a positive result. In hemagglutination inhibition, the laboratory turns a virus's own trick against it, because many viruses normally bind to red blood cells and cause them to clump together, but when a patient has been previously exposed, their serum contains specific antibodies that attach to the virus and block this binding. This illustration contrasts hemagglutination with hemagglutination inhibition, highlighting how viruses and antibodies influence red blood cell behavior. On the left, in positive hemagglutination, viral particles bind to red blood cells and cross-link them into a lattice, causing the cells to spread out and form a uniform red mat at the bottom of the well. On the right, in hemagglutination inhibition, specific antibodies bind to the virus, blocking its attachment to red blood cells. As a result, no lattice forms, and the red cells settle by gravity into a compact red button. This absence of hemagglutination provides strong evidence of immunity or past infection, turning the lack of visible clumps into a clear diagnostic signal. Now let's shift to another antigen-antibody interaction, the precipitation reaction. When soluble antigens react with specific antibodies in the zone of equivalence, insoluble immune complexes form and precipitate. These reactions are used to identify and quantify soluble proteins. Examples of precipitation reactions include single radial immunodiffusion, Auchterlonie double diffusion, and immunoelectrophoresis. In single radial immunodiffusion, graded concentrations of an antigen are placed into wells cut into an agar gel containing a uniform distribution of antigen-specific antibody. As incubation proceeds, the antigen diffuses radially outward through the gel until it encounters its antibody in the zone of equivalence, where insoluble antigen-antibody complexes form a visible precipitating ring. The size of this ring reflects the amount of antigen originally present, so that the square of the ring diameter is directly proportional to the antigen concentration, allowing precise quantitative determination of the antigen in the sample. Conversely, in the Australoni double immunodiffusion test, both antigen and antibody are placed in separate wells cut into an agar gel and allowed to diffuse slowly toward each other, forming visible precipitating lines where they meet in the zone of equivalence. When two antigen samples are tested against the same antibody, the pattern of these lines reveals their immunologic relationship. A smooth, continuous line of identity indicates that the two antigens share the same epitopes and are immunologically identical. Crossing lines of non-identity show that the antigens have no epitopes in common. And a spur pattern of partial identity appears when the antigens share some epitopes but one contains additional unique determinants, making this technique a powerful qualitative tool for comparing antigenic relatedness. Immunoelectrophoresis is a precipitation reaction commonly used to quantify serum proteins. A patient's serum is first placed in an agar gel and exposed to an electric field, which separates its proteins into distinct bands according to charge and size, including albumin and the various globulin fractions. A trough containing specific antiserum is then cut alongside these separated proteins, and as antibodies diffuse toward the protein bands, they meet their corresponding antigens in the zone of equivalence, forming visible precipitating arcs. Each arc represents a specific antigen-antibody reaction, allowing precise identification and qualitative assessment of individual serum proteins. We'll now discuss the labeled antibody systems, including the enzyme-linked immunosorbent assay, or ELISA, immunofluorescence, and radioimmunoassay. We'll start with ELISA, where enzyme-labeled antibodies transform invisible antigen-antibody binding into a measurable color signal. In the sandwich ELISA, a capture antibody is first bound to the plate and traps the target antigen from the sample. Then a second enzyme-linked detection antibody binds to a different epitope on the same antigen, forming an antibody-antigen-antibody sandwich, and when the substrate is added, a color develops. In contrast, the indirect ELISA is designed to detect patient antibodies. A known antigen is fixed to the plate, patient antibodies bind if present, and an enzyme-labeled secondary anti-human antibody reveals this binding through a color change, making ELISA a highly sensitive and versatile tool for both antigen and antibody detection. In most ELISA panels, the color intensity is directly proportional to the amount of antigen present because the detection antibody is conjugated to an enzyme that catalyzes the production of a colored product. When the substrate is added, each enzyme molecule catalyzes a chemical reaction that converts the colorless substrate into a colored product. Therefore, higher antigen concentration leads to greater enzyme activity and a deeper, stronger color, which can be quantified using a spectrophotometer. In fluorescent antibody testing, antigen-antibody interactions are visualized using antibodies tagged with fluorescent dyes. In the direct fluorescent antibody test, a tissue or clinical sample is treated with fluorescently labeled antibodies that bind directly to the target antigen, so the presence and location of the antigen can be seen immediately under a fluorescence microscope. In the indirect fluorescent antibody test, a known antigen is first exposed to the patient's serum, and if specific antibodies are present, they bind to the antigen. A second fluorescently labeled anti-human immunoglobulin is then added to detect this binding, producing bright signals that reveal whether the patient has mounted an immune response to the pathogen. Finally, in radioimmunoassay, a known amount of radiolabeled antigen and the patient's unlabeled antigen compete for a limited number of binding sites on a specific antibody, creating a highly sensitive competitive binding system. When the patient's sample contains a high concentration of antigen, it displaces more of the radiolabeled antigen from the antibody, resulting in lower measured radioactivity in the antibody-bound fraction. Conversely, low antigen levels allow more radiolabeled antigen to remain bound. By comparing the detected radioactivity to a standard curve, the exact antigen concentration can be calculated with great precision, though this powerful technique has largely been replaced by safer, simpler methods such as ELISA. From the microscopic fights involving intricate binding forces to the grand strategies employed in diagnostic tests, the tales of the immune defenders are a profound testament to the incredible complexity, precision, and adaptability of our immune system. Whether operating on the microscopic battlefield within our bodies or in the advanced diagnostic lab, the intricate and dynamic dance of antigen-antibody reactions ultimately decides the fate of disease, ensuring the continued health and victory of immunopolis.